Hello students and my dear friends. In this session, we'll discuss with one of the important topic, that is culture media. So what is culture media? So culture media, it consists of all the nutrients which are essential for the growth of the bacteria and fungi. So culture media is required to grow the organisms from infected material. Infected material is nothing but clinical specimen, which we collect from the patient to identify the organisms. First of all, we have to grow the organism. For that, we need the a culture media. Culture media, it consists of all the nutrients which are essential for the growth of the bacteria. And after identification of the organism, the culture media, it is used for antibiotic sensitivity testing. So, if you see the history of culture media, the original culture media, which was used by scientist Louis Pasteur, that is urine or meat broth. So meat broth and urine, it also supports the growth of the bacteria. If we use the liquid medium, in the liquid medium will get the diffuse growth. So if we get diffuse growth, it helps to it is difficult to identify the bacteria. That makes the bacterial identification difficult, and we can't study the exact properties of the bacteria. And if we use the a solid medium, so solid medium, discrete colonies are produced, so that we can easily study the properties of the colonies, such as colony morphology and hemolysis, or otherwise any pigment production like that. So what is meant by colony? So colony is the macroscopically visible collection of millions of bacteria originating from the single cell. What is called colony is the macroscopically visible collection of millions of bacteria originating from the single cell. So colony is visible. Colony, it is the visible. And cooked cut potato medium, which is the earliest solid medium that was prepared by scientist Robert Koch. Here, in the preparation of this solid medium, initially they used gelatin, but gelatin is not satisfactory because gelatin liquefies at room temperature. That is, gelatin liquefies at 24 degrees centigrade. So that's what gelatin is not satisfactory. See, these are the basic constituents of culture media. What are those? So first one is water. Water is the source of hydrogen and oxygen. And next we add electrolytes such as sodium chloride or other electrolytes, NaCl and other electrolytes are added. Next, peptone. Peptone, it is a complex mixture of partially digested protein. See, this peptone, it is obtained from the lean meat or heart muscle, and it is digested by proteolytic enzymes. And if you see the composition, so peptone, it consists of proteases, amino acids, polypeptides, phosphate, minerals, and growth factors. What are these growth factors? Such as nicotinic acid and riboflavin. So this is the composition, and these are the minimum nutritional requirements for the growth of the bacteria. See, even some special brands of peptone also available, such as neopeptone, protease peptone. They are for special purposes. And next, we add meat extract. Meat extract is commercially available with the name of Lablemco. And meat extract consists of protein degradation products, inorganic salts, carbohydrates, and growth factors. And yeast extract. So yeast extract, it is prepared commercially from baker's yeast, and it consists of amino acids, carbohydrates, and inorganic salts. Next, blood or serum. See, blood or serum, this is added for enriching the culture media. See, some of the bacteria, they are exact in their growth requirement. They are fastidious bacteria. Fastidious bacteria means they are exact in their growth requirement. So to grow that bacteria, we have to enrich the culture medium. We enrich the culture medium by adding of blood or serum. Okay, so like Haemophilus influenza. Here we add 5 to 10 percentage of defibrinated sheep blood is added. And in some cases, such as in case of low flux serum slope, we add serum also. And next, agar. See, agar, it acts as the like the solidifying agent. It was introduced by scientist Fanny Hussey. She is considered as mother of microbiology. Agar, it is used for preparation of the like a solid medium. And agar, it is extracted from the seaweeds. That is from like algae. That is Geladium species. This is obtained from the like Geladium species. And see, agar, it has no nutritive value. It doesn't affect the growth of the bacteria, and it is not affected by the growth of the bacteria. And agar, it melts at 98 degrees centigrade and it solidify at 42 degrees centigrade. It melts at 98 degrees centigrade and solidify at 42 degrees centigrade. And it has no nutritive value. It doesn't provide any nutrition to the bacteria, and just it acts as a solidifying agent only. Here, in the preparation of solid media, generally we use 2% of agar. And see, New Zealand agar, it has like twice jellifying capacity than the Japanese agar. Next, BHI broth, brain heart infusion broth. See, this brain heart infusion broth, it is used for culture of non-fastidious and moderately fastidious organisms. Non-fastidious means they are not exact in their growth requirement, and they are used in the cultivation of moderately fastidious organisms. So especially this is recommended for Streptococcus pneumoniae for bile solubility test. And BHI, it consists of brain and beef heart, peptone, glucose, sodium chloride, and buffers. And even it is used as blood culture media. So this it is also used for inocula for antibiotic sensitivity test. It is also helpful to prepare the like inoculum for antibiotic sensitivity test. Next, preparation of culture media. So how to prepare the like culture media? So generally dehydrated media, they are commercially available in the like powder form. So just we have to take this dehydrated media and mix it with the like water. So based upon our need, like we have to add some agar powder, like appropriate amount of agar powder is added to the water, and it is dissolved and it is sterilized by autoclaving at 121 degrees centigrade, 15 lb pressure for 15 minutes. And after that, this culture media, it is poured into petri dishes. It is transferred into petri dishes when the temperature is agar comes to the like 45 degrees centigrade, and keep it aside for like 20 minutes. Allow it to set for 20 minutes. So this is preparation of culture media. Next, types of culture media. So culture media, based upon their consistency, it is classified into 3 types: solid medium, liquid medium, and semi-solid medium. Next, based upon the constituents or ingredients, they are classified into 4 types. First one is simple medium. Second one, complex medium. Third one, synthetic or defined medium. And fourth one, special medium. And again, the special media, it is of following types: enriched media, enrichment media, selective media, indicator media, differential media, sugar media, and transport media. And next, based upon the oxygen requirement, it is classified into two types: aerobic media and anaerobic media. So this is based upon the presence of molecular oxygen and reducing substances in the medium. If you see the first one, based upon their consistency, first one is solid media. See, solid media, it consists of 2% agar. It is a solid in the state. It consists of 2% of agar. And what are the advantages of solid media? So we can study the colony morphology. So how is the colony, whether the colony is a circular or like a fimbriate, or we can study the colony morphology and pigmentation. So whether bacteria is producing any colors. So if you see Staphylococcus aureus, it produces golden yellow pigments. And hemolysis, so breakdown of RBC. So breakdown of the RBC also we can see. So examples, nutrient agar and blood agar. Examples, nutrient agar and blood agar. In the picture you can see this is the nutrient agar. Next, liquid media. So liquid media is prepared without adding agar. Okay. If you do not add agar, then automatically culture media will be in the liquid state. So this is helpful for inoculum preparation, for blood culture, and even for isolation of pathogens from the mixture. Example, nutrient broth. Next, semi-solid media. So how to prepare the semi-solid media? So generally, if we add 2% of agar, it will be the a solid media. Then just reduce the agar percentage, decrease the agar percentage. If you add 0.5% of agar, then it will be the a solid media. The purpose of this is solid media is to check the motility. So we can check the motility of the bacteria, that is movement of the bacteria we can check. Next, based upon the constituents or ingredients, they are classified into simple media and complex media, synthetic media, and special media. So if you see simple media or basal media, nutrient broth and nutrient agar. Nutrient broth and nutrient agar, they are examples for simple medium or basal medium. If you see nutrient broth, it consists of peptone and meat extract and sodium chloride. And see, to this nutrient broth, if we add 0.5% of glucose, it is called as glucose broth. And next, peptone water. So peptone water is also one of the simple media or basic media. So peptone water consists of peptone 1% and sodium chloride 0.5%, and that is mixed in the water. And see, when 2% of agar is added to the nutrient broth, then it is called as the nutrient agar. So this nutrient broth and nutrient agar, they are the simplest and routinely employed medium in the laboratory for diagnostic purposes. So as I told you, if the concentration of agar is reduced, that is instead of 2%, if we reduce it to the 0.5%, it will become the a semi-solid media. So what is the purpose of semi-solid media? So to check the motility of the bacteria. In the picture you can see, this is the nutrient broth and nutrient agar. Next, complex media. What is complex media? Media other than basal media called as the complex media. See, complex media have the special ingredients. They have added ingredients. So this helps to know the certain properties of the bacteria. It helps to know the certain properties of the bacteria, and they provide some special nutrients which are required for the growth of the bacteria. Next, synthetic or defined media. What is synthetic or defined media? The media which is prepared from pure chemical substances and their exact composition is known. So the media which is prepared by using of pure chemical substances, and the exact composition is known, that is called as the synthetic media. So what is the purpose of synthetic media? It helps for special studies such as metabolic requirements. So the best example is Dubose medium with the twin 80. Dubose medium with the twin 80 is the example for synthetic or defined media. This is helpful for isolation growth of the Mycobacterium tuberculosis. Next, special media. In that first one is enriched media. So what is enriched media? Substances such as blood or serum or egg added to the basal media. What is basal media? Basal media is like nutrient broth and a nutrient agar. So to the nutrient agar, if we add blood or serum or egg, that is considered as the enriched media. So what is the purpose of enrichment media, enriched media? It is used to grow the bacteria which are exact in their nutritional needs. Example, blood agar, chocolate agar, and low fluorescence serum slope. So if you see, this is the blood agar. So blood, it is added to the a molten nutrient agar at 45 degrees centigrade. So generally 5 to 10% of sheep blood is used. Sometimes we can use horse blood also. And blood agar is the most widely used culture medium in the microbiology laboratory. And next, what is the advantage of this blood agar? We can see the hemolysis. So hemolysis is easily observed on this medium. So this hemolysis, it is used to differentiate between a gram-positive cocci and other bacteria. So it can also be considered as a differential medium. So next, chocolate agar. So what is chocolate agar? In the chocolate agar, we add heated blood. This is heated blood agar for isolation of fastidious organisms such as Neisseria species and Haemophilus. So how to prepare this chocolate agar? This is prepared by adding of sheep blood, that is 5 to 10% to the molten nutrient agar at 70 degrees centigrade. This is by heating. So red blood cells are lysed in the blood, and the contents of the RBCs will be released, so that the color of culture medium changes to brown. So that's why it is named as the chocolate agar. And next, low fluorescence serum slope. So serum, it is added for enriching the culture medium. So this is helpful for isolation of Corynebacterium diphtheria. So next, enrichment media. So enrichment media is the liquid medium. Enriched media is the solid media. Enrichment media, see here we add some of the substances to the liquid culture media, and these substances, the inhibitory substances, are added to the liquid medium. So this inhibitory substances, they suppress the growth of unwanted bacteria, and they promote the growth of the wanted bacteria. So here enrichment media is the liquid media. So this liquid media helps to isolate the pathogens from the mixed culture. So here in the preparation of enrichment media, we add some inhibitory substances. What these inhibitory substances they do? They suppress the growth of unwanted bacteria, and they promote the growth of wanted bacteria. So this is examples: selenite F broth. So selenite F broth, this is helpful for isolation of Salmonella and Shigella. So if you see selenite F broth, it supports the growth of only Salmonella and Shigella. No other bacteria can be grown in the selenite F broth, because this is selenite F broth. It suppresses the growth of other bacteria, and it promotes the growth of Salmonella and Shigella. Next, alkaline peptone water. This is helpful for isolation of Vibrio cholera. It supports the growth of only Vibrio cholera, and it suppresses the growth of other bacteria. So next, selective media. So selective media, what is selective media? Here, the inhibitory substances are added to the solid media. In the enrichment media, the inhibitory substances are added to the liquid media. In the selective media, the inhibitory substances are added to the like solid media. So here also this media helps to isolate a particular bacteria from the specimen when mixed bacterial flora is expected. So here selective media and enrichment media. So both have the inhibitory substances. They suppress the growth of unwanted bacteria and they promote the growth of wanted bacteria. But enrichment media is the liquid media and selective media is the solid media. So example, TCBS medium, it supports the growth of only Vibrio cholera, and LJ medium, that is Lundsten-Jensen medium, it supports the growth of only Mycobacterium tuberculosis. Wilson and Blair medium, this is helpful for isolation of Salmonella typhi. Potassium tellurate medium, it is helpful for isolation of diphtheria bacilli. DCA, that is deoxycholate citrate agar. So here it is also helpful for isolation of only Salmonella and Shigella. And bile salt agar, so bile salt agar, here bile salt, it is a like selective agent. It also like supports the growth of only Vibrio culture. In the pictures you can see it is the TCBS medium which supports only Vibrio cholera, and DCA, deoxycholate citrate agar, it is helpful for isolation of Salmonella and Shigella. Potassium tellurate media, it is for isolation of Corynebacteria and diphtheria. And LJ medium, Lundsten-Jensen media, for isolation of Mycobacterium tuberculosis. So next, indicator media. So what is indicator media? Indicator media consists of an indicator. So what is this indicator? Indicator changes its color when a growth of the bacteria takes place. So indicator media consists of indicator. So they changes their color. So before the growth of the bacteria, the color of culture medium is different, and after the growth of the culture media, after the growth of the bacteria, the color of culture medium is changed. So example, McConkey's agar. See, McConkey's agar, it is both differential media as well as indicator media. So McConkey's media, it consists of indicator, that is here Enriched indicator it has. So here, due to the fermentation of lactose and due to the acidic pH, the pink color colonies are formed in case of neutral red indicator. So next, differential media. So differential media, it has substances incorporated in it. It is unable to distinguish between the two bacteria. So this media consists of substances which help to distinguish between the different characteristics of bacteria. So again, example is McConkey's medium. McConkey's medium consists of peptone, lactose, agar, neutral red, and terephthalate. So neutral red act as a indicator. So it comes under indicator media as well as the differential media. It distinguish between lactose fermenters and non-lactose fermenters. So lactose fermenters produce pink color colonies and non-lactose fermenters produce colorless or pale colonies. And one more culture medium is CLED agar, that is cysteine lactose electrolyte deficient agar. So this is for culture of urine specimens. This is alternative combination of blood agar and McConkey's agar. So it also differentiate between lactose fermenters and non-lactose fermenters. So what are the advantage of this medium? So this medium is less inhibitory than the McConkey's agar medium, and this cysteine lactose electrolyte deficient agar, it supports the growth of gram-positive bacteria and Candida species. So here you can see this is McConkey's agar. This is original culture medium, and here lactose fermenters produce pink color colonies and non-lactose fermenters produce pale or colorless or clear colonies. So here the color of culture medium is also changed. So it comes under indicator media as well as differential media. So next, sugar media. So what is the sugar media? Sugar media consists of like any fermentable substance such as sugar, such as glucose, arabinose, lactose, starch, etc. So what is the purpose of this sugar media? So it helps to check the like a fermentation test. The sugar media, it consists of 1% sugar in the peptone water, and it consists of small tube, that is Derham's tube. In the picture you can see it consists of small tubes, Derham's tube, that is kept inverted in the like larger sugar tube. So it helps to detect the like a gas production by the bacteria. And here it has like a Enriched indicator. So this Enriched indicator helps to check the like acid production by the like bacteria. So next, transport media. So transport media, this is used for transporting the like samples. So some of the organisms are delicate organisms such as gonococci or other bacteria. So these delicate organisms, they do not survive time taken for transporting the like specimens. Uh, in that cases, after collection of sample, the sample it is placed in the like a transport media. See, this transport media, it do not allow the bacteria to multiply, but it maintains the like viability. Here example, Storz medium. So Storz medium is the non-nutrient soft agar gel. It consists of a reducing agent. So what this reducing agent, it prevents the like oxidation, and charcoal neutralizes the bacterial inhibitors. So it is helpful for transporting the like gonococci. And next, buffered glycerol saline. This is a transport medium for enteric bacilli. VR medium, Venkatraman Ramakrishnan medium for Vibrio cholera. Next, anaerobic media. So anaerobic media, they are used to grow anaerobic organisms such as Robertson cooked meat medium, thioglycolate medium. They support the growth of anaerobic organisms. So anaerobic organisms, they do not require oxygen for their growth. If you see, this is thioglycolate broth. Thioglycolate broth consists of nutrient broth and 1% thioglycolate. Robertson cooked meat medium, it is also called as CMB, cooked meat broth. It consists of nutrient broth and like pieces of fat-free minced cooked meat of ox heart. Here, based upon the color change of the like meat particles, we can identify the growth of the bacteria. So if you see Clostridium perfringens, it turns the like meat pieces into red color. And in case of Clostridium tetani, the meat piece it is turned into black color. Next, media for growing of the like fungi. Here, SDA, that is Sabrod's dextrose agar media. So this culture media helpful for growing of the fungi. And SDA consists of high concentration of either glucose or maltose and mycological peptide. It has acidic pH, almost the pH of 5. Here we add chloramphenicol or gentamicin. So chloramphenicol, it is added to inhibit the like growth of the contaminating bacteria. This is SDA. Next, media for testing special properties such as urea media. So urea media is used to test the property of urease production. And PPA medium, phenyl pyruvic acid medium. This is helpful for identification of Proteus species. And dehydrated media, what is dehydrated media? So this is commercially available. So this dehydrated media simply reconstituted by adding of distilled water, and it is sterilized by using of autoclave. So what is the advantage of this dehydrated media? It makes the the process of media making become simple, and the quality will be the like more uniform. Here, composition and uses of liquid media. If you see peptone water, What is the composition? It consists of peptone 1%, sodium chloride 0.5%, and here we have to add it to the water 100 ml, and the pH should be adjusted to 7.4. What are the uses? Routine culture and for sugar fermentation test. Next, nutrient broth. Nutrient broth to the peptone water, add meat extract, that is 1% of meat extract. It is also for routine culture. Next, glucose broth. To the nutrient broth, add just 1% of glucose. This is for blood culture and even for growth of the streptococci. So blood culture and culture of some of the organisms, such as for culture of streptococci. Next, enrichment media, that is liquid media, alkaline peptone water. So here we have to prepare peptone water and the pH should be of 9.0. What are the uses for culture of Vibrio? Next, selenite F broth. It consists of peptone water and sodium selenate. This is for culture of feces for Salmonella and Shigella. Next, composition and uses of solid medium. Like simple medium, what is the simple medium? Nutrient agar. What is the composition of nutrient agar? To the nutrient broth, add 2 to 3% of agar. This is for routine culture. Next, enriched media such as blood agar. To the nutrient agar, add 5 to 10% of sheep blood. This is also helpful for routine culture. Next, to chocolate agar, heated blood agar. Composition is heated blood agar at 55 degrees centigrade for 2 hours. This is helpful for culture of Neisseria and Haemophilus influenza. Next, to low flux serum stove. It consists of nutrient broth, glucose, and horse serum. This is also helpful for culture of Corynebacterium diphtheria. Next, indicator medium. What is the composition? Peptone, lactose, agar, sodium taurocolate, neutral red. This is for culture of gram-negative bacilli. Next, selective media such as deoxycholate citrate agar. Composition is nutrient agar, sodium deoxycholate, sodium citrate, lactose, and neutral red. This is for culture of Salmonella and Shigella. Next, bile salt agar consists of nutrient agar and sodium taurocolate, and the pH is of 8.2. This is for culture of Vibrio. This is the end of the session. What is the summary? Culture media, it is required to grow the organisms from the infected material, that is from clinical sample. What are the basic constituents of culture media? Such as water, electrolytes, peptone, meat extract, blood, serum, and agar. So these are the different type of special media such as enriched media, enrichment media, differential media, indicator media, selective media. These are some of the special medias. Thank you.